All right, well, thanks to all of you that have been so faithful in coming to class. I appreciate you that I'm not just, talking to an empty space or a blank computer screen all the time. I think evaluations, maybe the link to that may have been posted. If it's not already, it should be soon. So I would encourage you to fill that out. I'm teaching in this same room, the same space next semester. So if you have any suggestions, I know I need to get a better mic. I'm going to work on that over break, but I would certainly welcome your feedback. I do read those, so try to fill that out. Obviously, we're going to be online from here on out after today. So what I'm planning right now is to just record those lectures and post them. I will have lots of office hours by Zoom, so don't be afraid to jump on there and discuss anything that you want to. So far, my Zoom office hours have been pretty quiet, so don't be afraid to do that. I'll be there and I'll post again what those office hours will be. Remember, your exam 4 is 1 week from today. So if you haven't used Respondus yet, there's a practice, just a couple questions, just kind of to make sure you've got everything that you need for that. I haven't really had the problems that we've had with that have been from my end. Like I've posted something incorrectly. So you would think by now that I've kind of made all the mistakes that I should have made, and hopefully it will go off without a hitch. But I would like everybody to take that together on Tuesday, 10 o'clock. You have till 11:15. If you want to take it before that, just make some arrangements with me. I'm glad to have you take it earlier than that if that would work out better for you. I will make some announcement after today about exactly how far the exam will cover. I'd like to have it cover at least through Staphylococcus, so we have like something on the exam that's not just immunology. So we should get into the Staphylococcus today, and I don't know, maybe even beyond. But I will be trying, as I lecture online, I'll try to, I will post lecture outlines, but I try to post ones that have a little bit less writing on them. So if you don't have access to a printer, you can pretty much write everything down. It is good to take notes rather than to just, scrolling through slides. It just engages you a little bit more. So when you actually do the writing, so that's, I think, in general, a good thing. Lastly, last announcement, the chapter 19 homework that covers Staphylococcus and Streptococcus, I have as due next Tuesday for your exam, because I don't think we'll get through all of Streptococcus, or if we do, I'll have to make some decision about what's covered. I may end up making the chapter 19 homework not due until the final. So that makes your homework assignment a little bit more manageable. By all means, go ahead and do the staff homework, Staphylococcus, because again, that will be on the test, but you can break it up and do part of it now and part of it later, the Streptococcus part later. So remember, homework due 10 o'clock there. I wanted to finally talk about a couple of questions from the last exam. I was trying to give a few students benefit of the doubt that I allowed them to take it a little bit late. So finally got everybody to take it now. There was really only a couple questions that more than half of you missed. And like, I think this one was like maybe 51% of you got it correct or something like that. This was kind of a big picture question about sulfa drugs. So SXT interferes with enzymes that are necessary to synthesize folic acid. Okay, why do bacteria need folic acid? Why do we need folic acid? That's in order to manufacture DNA. And if you look back on our slide here, this may have seemed like a detail, but this is the whole point of this pathway. This is the reason that we need folic acid in order to synthesize DNA. So it's kind of like the end game there. Another question, which of the following chemical agents has the distinct disadvantage of losing its activity when exposed to light? I think the problem with this question is many of you didn't recognize that sodium hypochlorite is bleach. Okay. In the past, we've always used sodium hypochlorite in labs to write down benches, but we didn't use that this year, so that might have been not as commonly recognized. It was in the notes. Hypochlorite is bleach, but I think that's probably why that one is a little bit more commonly missed. And one more about drug-resistant populations of microbes. How do they arise? Well, if you remember back to this graphic in your notes, how there are random mutations that cause a few bacteria to maybe have drug resistance. Typically, those organisms would be outcompeted. They might not be as fit to survive in most normal environments. They typically wouldn't survive unless they were had some selective pressure, unless they were in the presence of drugs where the other cells that they were kind of competing with would die off and only the antibiotic resistant ones would reproduce. So if you go back to this question, it's not the exposure to drugs that causes the mutations, just there are random mutations. And then when the organisms are exposed to the drugs, then it allows the overgrowth of the resistant cells. So that's the theory that we have anyway. Okay. I also wanted to take a minute. Our last lecture, like that was chock full of some more conceptually difficult concepts, definitely. And I kind of encouraged you to stop me and ask any questions. I know it's hard to do in this setting, but I wanted to give you a couple of practice questions about kind of that adaptive immunity stuff that we did last time. So during T cell and B cell maturation, T and B cells with receptors that recognize autoantigens are sorted from the rest of the B and T cell population. What do we call this process? This is the last thing that we did as we finished last week. This is a process called clonal deletion. So we're deleting out any B cell and T cell receptors that would recognize our self-antigens. Okay. All right, in a cell-mediated immune response, cytotoxic T cells, how are they activated? Okay, let me give you a chance to look at that just a second. Okay, and I think it's definitely helpful as you do multiple choice questions to scratch out the ones that don't make any sense. Okay, so I can throw a lot of stuff up here that just doesn't make any sense. Okay, this perforin granzyme pathway, that's what T cells do to kill cells. So that's not going to be it. It's actually, they have to have the release of cytokines from activated T helper cells. Okay, so remember, it's a special population. Once the T helper cells are activated, they differentiate into T helper type 1 cells to act on the cytotoxic T cells. One more. All right, it's a little more straightforward, a little more kind of just got a memory. All nucleated cells in the body have the ability to advertise endogenous antigen on the surface of their cells by means of dendritic cells. That doesn't make sense. B cell receptors, that doesn't make any sense. Facebook, no. Okay, so hopefully you could narrow it down to one of these two, and that's just kind of something you have to know. Remember, cells volunteer what is going on inside of them. So they raise their hand, one arm, that's MHC type 1. Okay, it's only the antigen presenting cells that have the MHC type 2 to present antigen that they have engulfed. All right, any questions about any of those before we move on? Yeah.
Do you plan on posting a lecture for Thursday with more material that will be on the exam, or is today the end of
Yeah, so question is, I do plan to post a lecture on Thursday. Yes, we will be moving. It kind of depends, obviously, how far we get today. Probably most of the Staphylococcus will be on Thursday. I'm thinking right now that I will probably not include that on the exam, but I will send something out to be more clear exactly where the cutoff will be. Okay. All right. All right, so let's move on. We're just doing a little piece of chapter 17 from your book, and that's the part that talks about immunizations. What an appropriate time to be talking about immunizations. I have so much to say today. I hopefully won't leave anything out. But I love posting this picture that you can find this on the web. I don't have a good citation for this, but this was put out in 1802 by the Anti-Vaccination Society. So even so that long ago, there was, pressure. There were people that didn't want to vaccinate. All right, we still see that today, right? Back then, the vaccination, the only one that they had was the cowpox vaccination that helped immunize people against smallpox. So smallpox, about 30% of the people that got smallpox died. So this was a really nasty infection, but Edward Jenner recognized that milkmaids that contracted cowpox from cows almost never got smallpox. So he took some scabs from a milkmaid that had cowpox and kind of ground them in, poked them into the skin of, a boy that he was testing it on, and let him get and contract cowpox. And then he intentionally inoculated the boy with smallpox by taking the scabs from somebody with smallpox and again stabbing them into the skin. And he didn't get smallpox. So that was the beginning of this vaccination. Vaca, that root comes from cow. But we didn't understand how it worked. And so anytime we don't understand how something works, then people tend to kind of be afraid of it and their mind wanders. So in this picture, it shows, did people really think they were going to get bovine characteristics from a cowpox vaccination? I don't know whether they really thought that or not, but we've got here a woman throwing up a cow or giving birth to a cow. This guy's got problems down here, sort of humorous, but it's just illustrative that, we can be afraid of things that we don't understand. And we certainly still see that today. So obviously, why do we want to vaccinate? In those terms, vaccination and immunization, we use those two terms pretty synonymous. We use them kind of interchangeably often, but technically, when you vaccinate, that means you get a vaccine. Okay? You get a shot, or sometimes vaccines can be oral. And immunization then is we're immunized when that vaccine encourages the proliferation of certain kinds of immune cells. Okay, so technically vaccination causes immunization. But again, we kind of use those terms the same. Okay, so why do you vaccinate? Usually a big priority is protecting children. We've seen how children have very poor immune responses to especially certain kinds of antigens. Kind of immature immune systems in general. So we want to protect children. Before we had vaccinations, about 1 out of 5 kids died. This was in 1900. About 1 out of 5 kids died before their 5th birthday. So we know that we want to protect children especially. We know that immunity wanes as we get older, right? So there's a number of vaccines that are recommended for adults as they age, as they get to be 60 or 65. The flu vaccination that older adults get is particularly robust. Okay? It takes a little bit more to kind of activate an older adult's immune system. But the whole point is that we want to get We want to try to reach herd immunity. Okay, and you probably heard that term in the news quite a bit. That's the idea that if most of the population is immune, then anybody that's susceptible is protected. Susceptible individuals are protected because there's just not enough people for the pathogen to, it's not likely, one person that's infected, they're not likely to come in contact with somebody else that could potentially be infected. So if we keep our herd immunity high enough, then the disease should basically go away. All right, so how high does the herd immunity need to be? Somewhere between 70 and 95%. I was just reading the other day that actually the herd immunity for influenza is lower than that. It's probably more like 30 to 40%, just because it's not terribly, even though it's respiratory. I'm not sure whether it's really considered airborne or not. It's just not terribly transmissible. Something like measles that is highly transmissible, we need to keep our herd immunity more like at 95%. So when we have people that kind of are lax about vaccinating their kids about measles, a year ago, that's all we were talking about, these big measles outbreaks that we had in New York and Disneyland, maybe a few years before that. So this is probably the biggest argument in my mind as to why people should be vaccinated. Again, not just kids, but adults. COVID, for example, because when a lot of people decide, well, I'm not going to do that, it's my choice, it's my body, I'm not going to do that, then that doesn't just affect them, it affects other people that can't be vaccinated. There are immunocompromised individuals where a vaccine would either be dangerous or it just wouldn't work. Young babies can't be vaccinated for some things because it just doesn't work when they're young. So we want to, as a society, protect those individuals that can't be vaccinated. Maybe somebody has an allergy and they're not able to tolerate the vaccine, or they had a bad reaction the last time they had a vaccine. So I think, to me, that's the strongest argument that for the general good, everybody should be vaccinated unless there's some health reason that they can't be. Okay, so it is hard to change people's minds about that without getting in an argument, though. I had one of my best friends, the topic of vaccination came up, and she said, well, I'm not getting the COVID vaccine. And it kind of took me back, and I was kind of surprised, but it didn't really matter what I said. She kind of had her mind made up. So, just when you're talking to somebody, decide whether it's worth an argument, decide whether it's worth a fractured relationship. But if somebody comes to you and wants to know about vaccines, hopefully, since you're in microbiology here, hopefully you will have at least some good information, some facts that if somebody's curious, you'll be able to help them understand. So we are going to talk about the COVID vaccine in particular. We'll get to that at the end of the lecture today. All right, so when we give vaccinations, what are we hoping that does in our body? We want to induce an adaptive immune response. Okay, so ideally we want to get memory B and T cells. Some vaccines will only give memory B cells. Sometimes that's enough. Okay, ideally we get our cell-mediated response in there as well. Okay, so generally speaking, we have good vaccines to infections where you get it once and then you're not likely to ever get it again. Because that means, when you've had an infection, you get good memory and you don't get it again. So things like chickenpox or measles, typically if you get it once, You always hear about an exception somewhere, but generally speaking, you don't get it again. So that's why the issue of can somebody get COVID a second time, it's a really important question, because if the answer is yes, then either we're not developing good memory from the first time we had it, or it's changing enough that our memory cells don't recognize it. So that's a really important question that we hope the answer is no. Once you get it once, you can't get it again. But there's some infections that, you know, they just change so much that it makes a vaccine highly unlikely. HIV, we don't really know now if that's ever actually going to be possible because it mutates so much. Of course, influenza mutates too, but much more slowly over years. So, you know, if we get a vaccination every year, we can kind of keep up with the strains that are in circulation at any one time. There are some pathogens that we just don't make good memory to them. So when you get a staph infection, you can get a staph infection over and over again because we just don't have, it doesn't elicit a really good memory, you know, response. So there's different reasons why we don't have vaccinations for everything, but of course we're gonna keep trying to develop those. Let's talk about the types of vaccines that are in use right now. There's really only 3 types of vaccines that are in use right now. We'll talk about some different strategies we have to deliver vaccines. Of course, the COVID vaccines that are coming out, these vaccines we don't have any in use of that type right now, so it's kind of an exciting thing. Probably the oldest type of vaccine is an attenuated vaccine. Okay, so that means we have some weakened pathogen that's going to elicit an immune response without causing severe disease in the individual. Okay, so that agent, that virus or that bacteria has been modified so that it can't cause disease. How we modify it, sometimes we grow it in some kind of cell type that's not its normal host. So for example, the Sabin polio vaccine, that is grown up in monkey kidney cells instead of human nerve cells, which it would usually target. So whatever, when we grow that in a different cell line, then enough mutations build up that it's not going to cause severe disease, but our immune system is still going to recognize certain proteins and have a good immune response to it. Sometimes we would grow that pathogen at a higher or lower temperature, again, just to cause some mutations. Or like the vaccine that we have to tuberculosis, it's called BCG. We've just cultured that so many thousands of generations that there's been some mutations that have just made it a little bit milder, and again, able to elicit immune response without causing that disease. Okay, so there are some real advantages to this kind of vaccine. It's a very natural, adaptive response. We get very good protection from an attenuated vaccine. We usually, and I'll add to that, we usually only need maybe 1 or 2 doses. The measles vaccine, for example, we just used to give 1 dose. 95% of people were immune after 1 dose of that. In the late 80s, we started giving people a booster to kind of catch that last 5% of people that didn't get good immunity from the first one. So usually, again, less number of, usually don't need boosters or fewer boosters for an attenuated vaccine. You could consider this an advantage that it can actually cause a mild form of disease, and that we call that contact immunity. So for example, if you're trying to vaccinate a remote tribe in the Amazon, and you get maybe half of the villagers vaccinated, if those villagers get kind of a mild form of the disease, they can spread it to other villagers that didn't get vaccinated and thereby get enough herd immunity in that population. So we call that contact immunity. If you think about some disadvantages to attenuated vaccines, I would say the first disadvantage is that it may be transmissible. So what I just talked about as being an advantage, that can also be a disadvantage. And in our society today, we would more likely call it a disadvantage, because if you get, say, the mild form of polio, for example, and then you're around your neighbor's baby and you pass it on to that baby, right, that could be really bad for the baby. So we don't want to actually be able to transmit it to somebody that's immunocompromised, for example. So generally speaking, we don't want that to happen. Some other disadvantages, typically we have to refrigerate because it's a live pathogen, or what you consider a virus live, you would say it's viable. Okay, so typically we have to keep them cold, so that can kind of make distribution a little bit more difficult to remote areas. Probably the biggest disadvantage here is the possibility of reversion. Okay, so we've done something to mutate this organism so it's not as infective. Well, what if it reverts back to its wild type form? So the story of why this is important involves the polio vaccine. The first polio vaccine, of course, polio was a terribly scary infection. Every now and then it would kind of assert itself, and especially, kids would get this in the 1950s, for example. Kids could be crippled or die from polio. So it was a very scary thing. So when we came out with the Salk polio vaccine in the 1950s, that was a great thing. Sabin, and you may have heard, like, if you ever watch Michigan football games, they always, in their little advertisement of Michigan, they always will show Salk. They kind of take credit. He did some of his research for the polio vaccine at Michigan. I think it was Salk. I get mixed up sometimes. Yeah, it was Salk. So he did some of his research at U of M, and then he went on to University of Pittsburgh and actually developed the vaccine. But the Salk vaccine was a killed vaccine. We're going to talk about those next. When Sabin came out with his vaccine, which was attenuated in the 60s, and it was cheaper and it worked better. So we switched to using the Sabin vaccine, at least in this country. It also had the advantage of you could administer it orally. So I know my youngest in 96 had, it was kind of nice. Okay, this is one less needle poke. She could just, they got her to drink it. So that was kind of nice. Some of you may have had that as well. But in the year 2000, the Sabin polio vaccine was given in the Dominican Republic and Haiti, and had about 20 or so kids where it reverted and they got full-blown polio. All right, so just because of the remote possibility of that happening, we don't use the attenuated version of that vaccine in this country anymore. At least we now go back to the Salk vaccine. You have to have more boosters, but again, it's safer. Even if it's a very low possibility that would happen, we don't want to make a previous healthy person obviously have the disease that we're vaccinating for. All right, so for that reason, we typically don't use an attenuated vaccine unless nothing else works. All right, so the MMR, measles, mumps, rubella vaccine, those are all attenuated forms. We've tried to make a measles vaccine from some kind of killed vaccine. It just doesn't work as well. And again, we have to have really high herd immunity for that one. This is just, you don't have to memorize what's what here. That's just an example of some, again, weakened forms of attenuated vaccines. All right. The other really, probably the most popular kinds, this is supposed to be a 2 here. Probably the most popular vaccines that we use now are some kind of killed cell, also called inactivated vaccine, because okay, our virus is dead or alive. We'll call them inactivated. Okay, so you have, say, the bacteria that's been killed. All right, so it's absolutely unable to replicate in the host. Right, you cannot get the flu from the flu vaccine. It's impossible. Okay, you can get it anyway, but it wouldn't be from the vaccine. Typically, you need more boosters to attain a good level of protection than what you would from an attenuated vaccine. And what kind of response do we see? Are we, knowing what you do about B cells and T cells, are you more likely to get a cell-mediated response or a humoral response? Because the pathogen is dead, it's very unlikely that our cells would take it up and present it with MHC class 1. Okay, so we're mostly going to get a humoral response here. Okay, it's going to be macrophages, dendritic cells that are phagocytizing that virus or pathogen, presenting it with MHC class 2, B cells endocytizing it. You're going to get mostly antibodies in response to this. Usually that's enough. Okay, so some examples: flu, Salk polio, rabies, pertussis. Okay, so a killed vaccine can be considered either whole cell, or sometimes they just take parts of that whole cell, kind of the most antigenic part. It's usually some protein on the surface of the virus, for example, that we would just use part of it. So how do we kill that whole cell? You can treat it with like gamma rays or UV radiation, sometimes chemical. Remember formalin, that's an aldehyde. Very minute quantities is all it takes in order to kill it. So a vaccine would have, again, just minuscule amounts of formalin. Sometimes you heat it up in order to kill it, kind of an obvious thing to do. The biggest problem with whole cell vaccines is that they tend to cause a little bit more inflammation. In particular, that's with a gram-negative whole cell. Why could that be? What do gram-negatives have that gram-positive bacteria don't? Something in their cell wall. LPS, lipopolysaccharide, otherwise known as endotoxin. Okay, the endotoxin is intact with a gram-negative whole cell vaccine. So an example of when this is a problem is for pertussis. Okay, this is a gram-negative Bordetella pertussis is the name of the organism. And for years, this was the only vaccine that we had for pertussis, whole cell. There were problems with mostly things like redness at the site of infection, pain, maybe a little fever associated with that. So of course, it's much better than actually having pertussis. But when your baby is cranky after they have a vaccination, then it makes the mother not want to go and get, the next set of vaccinations. So it's kind of a problem, although this provided pretty good protection. With a subunit vaccine, typically there's just not quite as much epitopes for our immune system to recognize. So typically maybe need more boosters, maybe 3 or 4 boosters to get as good a protection. But we do have less problem with inflammation. So for many years, for example, make sure I write this right, we used to give the vaccine DTP. Okay, that stood for diphtheria, tetanus, and pertussis whole cell. Probably a lot of you had that vaccine. But now, typically what's given is diphtheria, tetanus, acellular pertussis, so DTaP. Okay, so again, this stands for acellular pertussis. Much less inflammation, at the site of injection with that acellular pertussis. The drawback is it doesn't protect as well. So we're finding now that vaccine wears off after about 10 years. So babies get their maybe series of 4 or maybe more, I forget, DTaP injections, and then you really need another pertussis by the time kids get to be like 10 or 12. A few years ago, I read a case study about that, how kids were getting pertussis even after they'd been vaccinated. Pertussis is most serious in young babies. So, but again, the more people that have it, the more likely they're likely to pass it on to a baby. So, okay, questions there? All right, so just to kind of show you a couple of vaccines that we've talked about here, polio and measles. Let's not forget the big picture. Like, sure, there's certain people that might have a little reaction. Usually those reactions aren't serious. Where would we be without those vaccines? The incidence of polio, of course, has gone way down. We haven't had a case of indigenous or polio that was from somebody that hadn't left the country in this country since 1979. I think in 1993 was the last time that somebody brought it in. We are really close to eradicating polio. It's only in Nigeria, Afghanistan, and Pakistan now. So huge campaign. We're hoping to just wipe that off, and then we don't have to have polio vaccines anymore because it's not out there. It's not a zoonotic infection. We can't get it from animals. It's just something we pass around among humans. So big public health success there. And of course, measles, pretty much before, mid 1960s, when that vaccine became available, pretty much everybody had measles. So if you get to see your grandparents over Thanksgiving, ask them, do you remember having measles? Okay, my own parents both had it when they were very young. So it's usually an infection that people recover from, but there's a lot of complications associated from it, a lot of death, especially in more underdeveloped countries from measles still. So we need to keep up our herd immunity with measles. The last type of vaccine that's in use right now, there's a couple of infections that we have toxoid vaccines to. So these are infections where it's the effects of the toxin that are really causing the problem. So we've taken the inactivated toxin. So again, by some chemical means or by heat or something, we've inactivated, denatured that toxin just enough so that it's not going to cause disease. And we tend to get just mostly an antibody, a humoral response from these vaccines. So they're typically not long acting. Tetanus, right? We should get tetanus about every 10 years because it wears off. Okay, so tetanus and diphtheria. Diphtheria is not an infection that you probably have ever seen or know anybody. We've pretty much eradicated this, come very close in the US anyway, but diphtheria causes these pseudo membranes to form in the throat that essentially will suffocate somebody. So it's, again, very serious, scary infection that, again, we've had really good success in getting rid of. Okay, let's see. All right, so let's talk a little bit more specifically about some things in the news. And I did add a objective. So if you studying for the test, I put a couple objectives there. Number one, I want you to understand what these the vaccines that have come out that we're talking about in the news the last couple of weeks, how they work. And I want you to understand the testing process, a little bit about that, how we ensure that vaccines are safe. Again, so this is good information to share with people you know. So if you look at what kind of COVID vaccines are out there, the last time I checked, there was 187 COVID vaccines that are in some stage of development. About only 38 of those have reached clinical trials. So clinical trials means we're testing them on humans. All right, if they're in development, then we're still testing them in animals to see if they're safe in animals, to see if they have any protection in animals, and see what kind of side effects we see. All right, so there's a lot of possibilities, a lot of people worldwide working on this. Some of the different kinds of vaccines, there's all different kinds of vaccines that are out there. Some of them take an inactivated virus. Some of them are considered subunit. So again, that's just a killed acellular, just part of the vaccine. And usually we are trying to take part of that, the spike protein. There's really only 4 proteins in the coronavirus. There's the capsid protein, there's a membrane protein, the spike protein. Of course, that's the one on the outside, so it's the one that our immune system is most likely to recognize. It needs that spike protein in order to invade cells. I'm missing one, I forget what the other one is. Envelope, envelope must be 2 in the envelope. Okay, so most of these vaccines are targeted against that spike protein. There are some that are considered viral vector vaccines. So in this case, that means you use some altered virus that can't cause disease. Seems like there's several vaccines that are using an adenovirus, which is a virus that causes a cold, a common cold. They're taking an adenovirus of chimpanzees, so a common cold that's even specific for, different hosts than us, and using that they're transforming that a little bit in order to make the COVID proteins, a spike protein. So it's just the spike, it's just a way to deliver that spike protein in a more natural way in order to elicit an immune response. And you might have heard of the Oxford vaccine. That's one of the 5 or so that we're hearing more about. That's the mechanism that one uses. And then there are RNA and DNA vaccines. And this is old technology. It goes back about 30 years ago. It was interesting to read about. There's a Hungarian woman working at University of Pennsylvania that was trying to get grant money to work on, she was trying to make drugs RNA drugs that would, teach somebody wasn't able to make certain kinds of proteins, a way to help somebody make their own proteins. And she had a hard time getting funding. She had all sorts of hurdles to get through because when you deliver that RNA, it typically induces an immune response just to that RNA. So there was hurdles to try to get our immune system not to react to that foreign RNA that was introduced. But it's a great idea, able to make some kind of protein in your cell, and then we generate an immune response to that protein. So the 2 vaccines that you've heard about in the last couple of weeks, last week, the Pfizer, again, these are RNA vaccines, came out that it had about 90% efficacy. So that's really exciting. We are really only hoping for a vaccine that would be at least like 70%. So 90% is really quite exciting. Just yesterday, I don't know how many of you caught it, but that Moderna vaccine, it's another mRNA vaccine, different chemical structure of the mRNA, different ways of getting it into the host cell. So there's slightly different vaccines. They're saying 94.5% efficacy. So that's really exciting. We have now a couple of vaccines that have been given to over 60,000 people so far. So we have an independent agency that's checking the numbers here. So we know that they're not skewing their data. So it's not been peer reviewed, but this is really good, exciting results so far. So one of the drawbacks with this Pfizer one, they're saying that it has to be kept at about minus 80 degrees Celsius. So that's a deep freezer. So you're going to have special ways to transport that. You got to be careful. Can't leave it out really at room temperature very long before you administer it. So you're going to have ways to check to make sure that that temperature has been maintained. Exciting thing about this Moderna vaccine is that it can be kept in the refrigerator. So that's a really big advantage in terms of trying to make sure that it hasn't lost its efficacy. Okay, both of them, you're going to need 2 doses about 3 to 4 weeks apart. So, you know, if you're trying to vaccinate 50 million people, you actually need 100 million doses of vaccine. So, but again, I think it's probably worth it. Okay, we don't really know how long the protection is going to last here. Usually when vaccines are tested, like they're not approved unless they protect for a year or 2 at least. So obviously we don't know that yet, but we're hopeful that they're going to be good. Already we're starting to see, you know, some rumors and myths about these vaccines show up on Facebook or whatever. An RNA vaccine cannot mutate your DNA, right? We've talked enough about genetics in here that hopefully you can help display that, you know, put the kibosh on that myth, but we don't have enzymes that make DNA from RNA. All right, so that can't happen. This RNA is actually, it only lasts a couple hours in our cells before cellular proteases will start to degrade it. So it's really only a very small amount of protein. And again, hopefully that's enough to make, again, the spike protein to make a good immune response. Okay. So far, they have not seen serious side effects. Maybe 10% of the people feel achy for a day or so. You got some redness around the site of infection, but again, very few serious side effects. Okay, and this is the link to where I got that information. So if you want to look that up a little bit more carefully, I found this really nice, it's a little bit blurry there, it looks like, graphic in an article. Again, I have the citation down there of what is really going on. So here, with an RNA vaccine, we're not actually, it's not clear exactly how that gets taken up into the cell, but it gets taken up into the cell and of course translated at ribosomes. There's some processing that we know takes place all the time within our cells, and some of that, our protein in our cells gets presented by MHC class 1 to cytotoxic T cells. And of course, these are the cells that we really want that's going to help stop a viral infection. Now, we know that cytotoxic T cells, they don't get activated to do, you know, secrete their perforin and granzyme on infected host cells without some T helper activation as well. So they don't show that in this graphic. I talked to an immunologist, Dr. Renkema, and she said that there's actually some T helper cells that get activated. We actually present some MHC antigen by MHC class 2 as well, which it's just sort of beyond the scope of this class. So there is some of that that can happen as well. Of course, with a subunit vaccine, then we are going to get both T helper cells and CD8 cells. So that shows here some T helper cells getting activated as well as B cells. So Again, that's with a subunit. All right, so how do you know that the vaccine is safe? So what kind of channels are we go through in this country? Question.
Did that process start? Like, I was still confused by that.
Right, so I started right here. So this is talking about 3 different vaccines: a DNA vaccine, an RNA vaccine, or a subunit vaccine. So it's got like 3 different vaccines all in the same graphic there. So I started with, hey, let's say that an RNA vaccine is injected, it gets taken up into our cells, and then protein made at our ribosomes. Okay. Okay. All right. So what kind of channels does a vaccine, what kind of hoops do we have to jump through in order to get a vaccine that's safe? I got this, I'm just posting part of this graphic. This is from the CDC's website of how a vaccine gets licensed. Okay, so in terms of clinical trials, it goes through 3, sometimes we say 4 phases. Of course, first you have to have preclinical, where you have the testing in animals that I mentioned before. Okay, so we had a lot of animals that have helped us in this process for sure. Okay, so you have more of an exploratory phase first, where you're just, again, testing in animals, making sure that there's not too many adverse effects in animals, making sure that it works in animals. And then when you start your clinical trials, you have a phase 1 where you have a very small group of people, less than 100, and you're monitoring them very closely after they have the vaccine to see if it's safe. And again, we haven't, even though we've gone warp speed, we haven't skipped any of these steps. To try to go a little bit faster, we've sort of overlapped these. So we started phase 2 before phase 1 was totally finished. That's my understanding. But in phase 2, then you're giving it to maybe hundreds of people. And you're kind of messing with the dose a little bit. So you're giving different people different doses. Of course, there's placebos thrown in there as well for some volunteers. So it's a blind test. Again, an outside agency is looking over the data, so it can't be doctored. And then if everything's, seems pretty safe, if standards for safety, efficacy, then you move on to phase 3, where I don't know if there's a set number. I've heard the number either 2,000 or 3,000. You have to give it to at least that number of volunteers. Again, both the Moderna and the Pfizer vaccine have been over 30,000 people. So again, at least 60,000 people have had this. And then they have to be observed for 2 months, because typically the adverse side effects, the really bad ones like seizures or something like that, are going to show up within the first 2 months. Okay, so again, we haven't, now that 2 months is over, that's when now they're starting to report, how good it lasts. Okay, and then after, so everything looks good in phase 3. It seems to work well. It has very few serious side effects. So after the FDA approves, then we have what's usually considered phase 4. And that just means that it's a monitoring system after people in the general public are given the vaccine. So if you ever go to the doctor to get a vaccine, they will typically give you a sheet of paper, talks about, hey, these are maybe some of the side effects that you can expect. If you have these side effects, call this number. Okay, it's actually a system called VARS, and that stands for Vaccine Adverse Effect Reporting System. All right, so we have this mechanism in place where if people are having serious effects, that gets reported. and potential for the vaccine to lose its FDA approval. Okay, so even after it's approved, we have mechanisms in place to make sure that it's safe and effective. We want, of course, the benefits to outweigh the risks. So vaccine have only gotten safer as time has gone on. Okay. Do we have questions? Anything about the vaccine? Anything you're wondering about? Yes. So years and years, like whenever it's being used. So even vaccines that we've used for a really long time. Yeah. Totally can't hear you. Oh, what did VAERS stand for? Again, Vaccine Adverse Effects Reporting System. Okay. All right. Well, let's move on. I'll try to keep you updated if anything else big comes along on the vaccine front, but definitely encourage the more people that get vaccinated, the better. So I'll definitely encourage that. Okay, so the rest of the semester, a lot of students generally enjoy this material that we're covering at the end of the semester here. We like to call it our bug parade. Basically, we're talking about a lot of medically interesting bacterial and viral infections. So I like it because we get to kind of pull in stuff that we've talked about all semester. So it's kind of a little bit of a review as well. And we're going to start, and I think the reading in your book, I have listed page numbers, but you're only going to be responsible for the pathogens that we talk about in class. So there's some other ones in chapter 19, don't worry about, again, just what we do in class. But we're going to start with our gram-positive bacteria, our cocci. And there's really three major groups of gram-positive cocci that cause disease in humans. And we're going to be particularly concerned with the Staphylococcus and the Streptococcus. Okay, so if you're in lab, you will, a lot of this will look familiar because you will have talked about it in lab. I am going to just briefly mention Enterococcus, but Enterococcus is very similar to Streptococcus. And so we're going to kind of really not focus too much on this. Okay, so starting with the Staphylococcus, you think about some characteristics of this genera. There are more than 40 different species in this genera. So we talk about 3 of them in lab. They're the 3 kind of most famous ones, the most clinically interesting ones, but know that there are a bunch of other ones out there. Okay, so we see them when we look at them through the microscope, we see them in grape-like clusters. So remember, I didn't post this because we did this a long time ago. Remember your different cell arrangements that you see for cocci. Diplococci, you can find them in chains, which we'll see in Streptococcus, but not Staphylococcus. Staphylococcus are typically in these grape-like clusters, all right, big clusters. And again, if you're in lab, you've probably seen that. Where do we find Staphylococcus in general? Very common to find them on us, all right, our skin, mucous membranes, upper respiratory tract to some extent, even places like our hair. Okay, they are in the environment as well. What are some of its major characteristics? Well, they are catalase positive. So if you're in lab, that should look pretty familiar. Remember, catalase is one of those detoxifying enzymes against toxic oxygen products. So the reaction that it catalyzes is that it hydrolyzes hydrogen peroxide into water and oxygen. Okay, so let's see, I can balance that. So when you do a catalase test in lab, you add your organism to the slide, you drop a hydrogen peroxide, and then you look for these oxygen bubbles as a result of a positive result. Okay, so pretty easy test to do. And of course, our Staphylococcus have catalase. Our Streptococcus don't. So it's a really nice test to tell those apart. Some of our Staphylococcus form capsules, usually kind of a wimpy capsule. Even so much that it's, when you do a capsule stain, sometimes you can see it, sometimes not. So maybe you'd consider it more of a slime layer, oftentimes. They are facultative anaerobes, so pretty flexible with their oxygen requirement, pretty flexible metabolically. And they have a little bit higher environmental resistance. So not so much as like endospores, or not as much as mycobacterium with their acid-fast cell walls, but they're generally a little bit tougher in the environment than, say, Streptococcus. Streptococcus is really wimpy. Okay, so they withstand temperature fluctuations. They are very tolerant of high salt concentrations, pH fluctuations, all right? They're just a little bit tougher. They can withstand that sort of thing a little bit better than some other organisms. So if we're going to focus mostly on Staphylococcus aureus, right? So that's the staph that really causes a lot of different infections. So we're going to look at some of the virulence factors and some of the infections that Staph aureus can cause. All right, so if you look at its kind of occurrence and how it spreads, it actually can be part of our normal microbiota. So, you know, if you have it in low enough numbers, then it's not going to cause an infection. Experts disagree about this, but it's usually considered an opportunistic pathogen because the infectious dose is greater than 1 million cells. All right, so the Staph aureus really has to kind of overwhelm that portal of entry, overwhelm our innate immune system in order to be able to establish an infection. Now, of course, if the host is immune compromised, then it's going to take less cells than that. Okay, so again, if the right opportunity arises, it's probably going to be, you know, more successful in establishing an infection. We see spread in both hospitals and in the community. In the hospital, you have to be really vigorous about disinfecting fomites. You know, anything like, you know, doorknobs and bed rails and things like that lots of people might be touching. Implants, that's a really problem. It forms biofilms on implants. So things like catheters or, you know, artificial joints, something like that is a real problem and makes it hard to treat with antibiotics alone when it's in our body like that. Community, it's a lot of skin infections that staph cause. So places like locker rooms or, you know, wrestling mats or something like that. Whenever people are in close contact, it's more likely to spread. So some of the virulence factors, remember, these are tools that Staph aureus has to be successful in establishing an infection. A couple structural ones, I mentioned that some staph are able to form capsules. And in this picture, it's a transmission electron micrograph of some Staph aureus that's been treated with some antibodies, so you can visualize the capsule a little bit better there. Of course, that capsule production makes it harder for our phagocytic cells to phagocytize cells, especially apparently neutrophils have a hard time. Apparently macrophages are a little bit more successful in phagocytizing a staph cell, even with the capsule. But the capsule does increase its ability to form biofilms on some kind of, you know, artificial surface, like some kind of implant. And here's one that we haven't talked about before, protein A. Okay, so Staph aureus, or I should say many strains of Staph aureus, has this protein A, okay, all around the surface of the cell. And what that protein does is binds the FC region of the antibody. So remember our antibody structure, the stem region is the FC, And the region up here, that's the part fragment of antibody binding. This is kind of the business end of the antibody. Oops, light change here. So the bacterial cell does not want the FAB portion to bind to it, because that's the part that's going to, maybe neutralize any adhesion proteins or block any adhesion proteins or cause complement activation. Okay, so that protein A binds to the FC region so that the business end can't do any harm to that cell. So a good analogy is, if you're going to catch a snake, you don't come at it head on, right? You grab it by the back of the neck. So, if this is a snake here, right, you grab it by the back of the neck so it can't bite you. All right, that's essentially what protein A does. It grabs a hold again of that stem region. So this part isn't doing any harm to this cell anyway. It could still bind to another cell. Okay, so again, we call that, it's a structural protein. All right, lots of exoenzymes that are potentially secreted, and these could be, other staphs could be able to secrete these as well, but again, we're talking specifically about Staph aureus, and some of these you'll recognize again if you're in lab. But coagulase, this is an enzyme that allows the organism to clot blood. So basically, when it infects a wound, then it can kind of wall itself off from our neutrophils, our macrophages that would be coming to try to phagocytize it. Okay. Again, it helps. So this is going to, a virulence factor that's going to help the Staph aureus establish itself in the host. Okay. Now, when the time is right, when maybe all the nutrients get used up in that little clot, then the organism, to invade elsewhere, is going to need to break that clot. So it also have to produce a kinase, and we call it staphylococcus. Okay. So dissolves the blood clot. And I should mention, right, if you're in lab, inoculate some rabbit plasma to see if your organism makes coagulase. You see any kind of a little clot there after you incubate it, then that's a positive. Pretty much, there are a few other organisms that make coagulase, but it's Staph aureus is kind of the one that's most famous for making that. Hyaluronidase, I have trouble saying that one. This was one that we mentioned back in chapter 14 as being an example of a virulence factor, of an exoenzyme. But remember, this that hyaluronic acid is kind of the matrix that holds our cells together. So it breaks down that matrix between cells. So helps it to be more invasive. Lipase is an exoenzyme that typically we test for in lab. It was one of the labs that we had to skip this year. But of course, lipase is an enzyme that hydrolyzes or breaks down lipids. Lipase activity is typically really closely correlated with pathogenicity in Staphylococcus, because it enables the organism to break down some of the oils on our skin that would maybe help it infect, say, a hair follicle, for example. And then beta-lactamase. There was some confusion about this on the last exam, but these are enzymes that bacteria produce that are going to inactivate beta-lactam antibiotics. Okay, so we have a huge problem with resistance for Staphylococcus. So we are having less and less all the time arsenal of antibiotics that are going to work against Staphylococcus because they acquire the ability to make this enzyme usually through some kind of horizontal gene transfer, like conjugation. Okay, so they acquire the plasmid that enables to make this enzyme that enables them to break that bond in beta-lactam antibiotics that keep them from working. All right, there's also an array of exotoxins that Staph aureus can make. Again, you should recognize this hemolysin if you're in lab. Now, we talked more specifically about hemolysins in Streptococcus, but it turns out that Staph aureus is actually beta hemolytic. Okay, it is able to lyse red blood cells. So typically, the only way that you know you have Staph aureus in lab is if you see a positive coagulase test. But actually, if you look at that organism on blood agar, you should see this yellow clearing around the growth for Staph aureus. And we typically see this in vitro effect. So that means in the lab, when you're growing cells on media, you can see this hemolytic effect. We actually think that when it's in our body, that you would think if it broke open our red blood cells, that would be a huge problem. We don't see that much effect actually in our body, thankfully, I guess, for whatever reason. I don't really know why. Okay. Enterotoxin, okay, not endotoxin, enterotoxin. Remember that prefix entero means intestinal. Okay, so some Staph aureus produce a toxin that acts especially in our small and large intestine, and that gives us the nice effects of nausea and diarrhea, vomiting. Okay. An exfoliative toxin, this is one where it causes our skin cells to separate and pull away. Okay, so of course we buy products that are supposed to be exfoliative, so to kind of get rid of our dead skin cells to make our skin look more vibrant. This goes beyond that, and you're losing your skin that, remember, is your kind of first line of defense. So that is a huge problem when your skin's falling off. We'll see an infection where that exfoliative toxin is. You can see what effect it's having. And this toxic shock syndrome toxin, we've mentioned this a couple times already. Remember, this is a super antigen. This toxin, remember the thing about toxins, it only takes exotoxins. It only takes a little bit of them, and they can have just wide-ranging systemic effects in the host. So because this is a super antigen, lots and lots of our T cells are being activated rather than maybe just a few of them. So we get lots of inflammation as a result of that. And we'll look at that toxic shock syndrome in a minute, but just massive cytokine production there from the super antigen. Right. So I know we're almost out of time here, but just starting to look at some of the infections caused by Staph aureus. We start with a lot of skin infections, and then there are some infections that kind of can really, your whole body can be impacted. But one of the probably most innocuous ones is folliculitis. So that means when a hair follicle has gotten infected by, again, Staph aureus. Usually it's more likely to happen if your skin has been if it's a little bit irritated already, maybe by shaving, or maybe somebody that has eczema, or maybe just wearing tight clothes. So maybe the skinny jeans aren't such a good idea. But it's an infection that looks a little bit pussy. So remember, some redness there. You can see the inflammation. The pus is just lymph along with some dead cells, immune cells, white blood cells mixed in there. Typically not something that's going to go more invasive. Again, kind of a superficial kind of infection. If it does get a little bit more invasive, oh, I lost some of my writing there. It can cause a furuncle or a carbuncle. Again, this is when the organism has got some more virulence factors to break down the tissue a little bit more. Another name for a furuncle is a boil. Typically, these are still going to heal up on their own, but you got to keep an eye on any skin infection that's not clearing up kind of within a reasonable amount of time. Carbuncles are typically, again, more ulceration, getting deeper into the skin. They're most often show up on the back of the neck. I don't really know why. But this is starting to get more serious. A physician might need to lance them and drain them. Definitely going to want to be on antibiotics to get rid of something like this. All right, so we will continue with the staph and hopefully maybe finish up the strep next time. Look for an email again about how much that exam will cover. I will be in my office for a couple hours this afternoon at least, so come see me if you have any questions. Have a good Thanksgiving and have a good day, I guess. All right.